So today we are going to see the remaining part from your pre-formulation and tablet. So again there will be one more session on pharmaceutical excipient, because this pharmaceutical excipient everybody used to ignore, but you may find few questions which are very important from this pre-formulation or simply pharmaceutical excipient. So whatever excipient we are using as a diluent, as a lubricant, as a disintegrant, so all these excipients, their characteristics. So these are also important, and this is also finding very important place in the question paper. So today we'll see different pharmaceutical excipient. So what is not already discussed. So whatever we have seen in the tablet, so out of that few other things are important, so I have enlisted it, and today I will try to take as much as I can. This is not a thing that anybody can cover in a one lecture, but I'm trying my best. Okay, again another lecture will be on same thing. So excipient, we already seen this. Excipients means what? So while making a dosage form, while designing a dosage form, say for tablet, capsule, or any other dosage form, so along with the API, active pharmaceutical ingredient, along with your drug, you need some other agents. They are known as the diluents. They are not therapeutically effective, but they are used as additives, adjuvant, to enhance the either bulkness, okay, to increase the binding property, to increase the disintegration, to increase the aesthetic value. Aesthetic means smell, okay, then color, then flavor, etc, etc. So excipients, now pharmaceutical excipients, they are added in a tablet dose form or in any other dose form. Please try to mute yourself. So excipients, they are added either to enhance the organoleptic properties or to enhance the aesthetic value, right? So in that perspective, you are adding color to the formulation, you are adding some flavors, you are increasing the sweetness. So to increase the palatability of that formulation, you are adding sweetener or sweetening agent. Okay, now to stabilize the formulation, you are using stabilizers. So various kind of stabilizer, for example, to prevent the microbial growth, you are using preservative. To prevent the product from oxidation, like oxidation due to air, oxidation due to light, oxidation due to free radical or microbial spoilage, so antioxidants, you are using emulsifier, you are using suspending agent in the suspension, you are using emulsifier in the emulsi emulsion and other creams, ointments, and topical dosage forms. Now, to enhance the dose accuracy, if your dose of drug is very less, for example 1 or 2 milligram, so you cannot formulate that 1 or 2 milligram drug with a in a particular formulation. So you have to increase the bulkness of that formulation. So for that you are adding diluent. है ना, you cannot form a tablet of 1 milligram. 1 milligram का tablet वे कैसे करेंगे, उसको पंच कैसे करेंगे? So you have to add dilutes, you have to increase the bulk. So diluent, bulking agent, then fillers. So these are the important for to increase the dose to or to effect the dose accuracy. Again, you are adding processing aids to aid the process. Okay, so to aid the process of granulation, you are using binder. To aid the flow of granules from the hopper to the die cavity, you are adding lubricants. Sorry, to run a machine smoothly, you are preventing the friction between parts of machine, you are adding lubricants. इसके ऊपर GPAT 2023 में क्वेश्चन आ चुका है. To improve the flow of granules from hopper to the die cavity, you are adding glidant. Okay, so to prevent the dust, you are adding anti-adherent. And to increase the release of drug, so different additives are used. For example, to effect the disintegration, you are adding disintegrant agent. To increase the bioavailability absorption, you are using penetration enhancer. To modify the drug release, you are using coating agent. So there are different type of excipients which are used in the dosage form, any kind of dosage form, say for example tablet solid dosage form, capsule solid dosage form, and other liquid dosage form also. So along with the drug, whatever is added, that is known as diluent, and that has some purpose. So either it has a organoleptic compliance purpose to increase the aesthetic value, to increase the palatability. So it may have stabilizing purpose like preservative, antioxidant, emulsifying agent, sustaining agent, to increase the bulkness like a diluent, and then fillers, etc., to aid the process like binding agent, lubricant, glidant, and anti-adherent, to increase the drug release, disintegration, penetration enhancer, coating agent. Okay, so these are the excipient. They are known as excipient. Okay, based upon their objective of addition in dosage form. इसका रोल क्या है भाई डोसेज फॉर्म में, है ना? कलर का रोल क्या है कि कलर में टैबलेट आपको अच्छा दिखना चाहिए, है ना? अच्छा दिखेगा तो आप लेंगे, अदरवाइज नहीं लेंगे ना, लाइक दैट. So the excipient based upon their objective of addition in the dosage form. Why that particular, why we are using cross carbonyl sodium? Cross carbonyl sodium is added because of the disintegration, right? So why you are using antioxidant to prevent the oxidation, right? So excipient classification based upon the objective of addition in the dosage. Now a pharmaceutical excipient should have criteria, particular properties, particular specifications. So what are the criteria for excipient used in the tablet dose, for example? So all excipient must not be toxic, and they are acceptable by all regulatory agencies where the product is going to be marketed. Suppose if you want to make a tablet, आपको tablet बनाना है, वो tablet आपको मार्केट करना है इंडिया में. तो इंडिया का जो रेगुलेटरी एजेंसी होता है आपका CDSCO, सेंट्रल ड्रग रिसर्च कंट्रोल ऑर्गेनाइजेशन, so that you have to that excipient should be passed by CDSCO. So it must not be toxic. It must be acceptable to the regulatory agencies of respective countries. If you where you want to market the drug, so if you want to market the drug in the USA, so you have to certify each and every ingredient, right from the API, excipient, process, plant, labeling condition, according to the US FDA. If you want to launch your product in the US, if you want to market your product in the US, so it must be non-toxic, acceptable by regulatory agency of countries where you want to market the product. Right now, it should be available, easily available, and it should be easily available and acceptable grades commercially. So whatever grade you want to use, that should be available. So it should be available in acceptable grades commercially in all countries, right? The cost of this excipient must be low, and they must be physiologically inert. इनका कोई रोल नहीं होना चाहिए. It should be inert physiologically, and they should be with acceptable microbial load. So there is a always okay, contamination in the excipient. Say for example fermented product, so in that अगर fermented product है dextrin, तो उसमें contamination आ सकता है fermentation bacteria का. So that should have acceptable microbial load, right? And must be contraindicated. Okay, must be contraindicated. Okay, if it must not be contraindicated in a nature of excipient. For example, sucrose. So if you are using excipient sucrose, so obviously that product is not suitable for a diabetic patient. So must not be contraindicated in the nature or because of its component. जैसे for example sodium bicarbonate, अगर एंटासिड है उसमें सोडियम भी है कार्बोनेट में, अगर कोई हाइपरटेंसिव पेशेंट है तो ऑब्वियसली उसमें कॉन्ट्राइंडिकेटेड होता है कि लेवल बढ़ाएगा सोडियम का, पेशेंट का ब्लड प्रेशर शूट हो जाएगा. Am I audible to you clearly? Audible है ना? So it must not be contraindicated by the nature. That is, for example, sucrose in case of diabetic, and must because of the its nature. For example, sodium bicarbonate in case of antihypertensive patient. Then the excipient must be stable physically as well as chemically. If your drug falls in the food category of food, if your drug is under food category, say for example, जैसे आज का विटामिन सी, they are not considered as a drug now. They are considered as a they are considered as a food only. Okay? So if your drug is in a category of food, then excipient must be approved. Whatever excipient you are going to use, that must be approved for food additives by respective agency. It must be color compatible, you know, The additives whatever you are using that must be color compatible. नहीं तो प्रोडक्ट का आपका कलर है येलो, आपने कुछ एडिटिव्स यूज किए, वो बन गए सब ब्लैक, नहीं चलेगा. Okay? And must not have undesired effect on the bioavailability of the drug product. Okay, so these are the few qualities of excipient that is required. Okay? If you are using excipient in your formulation, so these are the qualities of excipient that must have. We have already seen tablets. Okay, we have seen the pre-formulation. In that pre-formulation, so one of the parameter is nothing but the salt formation, right? Salt formation. Why salt formation? So you might have seen drugs, they are rarely present in their original form. Say for example, ephedrine. No, ephedrine is alkaloid, so it is sympathomimetic, but it is always given in the form of ephedrine hydrochloride or ephedrine sulfate. Now, why salt formation is important? Okay, to enhance the physicochemical properties of drug. Suppose, no, you know that very well. We have seen the alkaloids. So pre-alkaloidal bases, they are soluble in the organic solvent, but they are not soluble in the aqueous solvent. But for formulation purpose, you want to solubilize. If you want to prepare a ephedrine hydrochloride cup syrup, so obviously that ephedrine must be soluble in the vehicle, right? But as I told you, pre-alkaloidal bases, they are soluble in the only organic solvent. So you have to convert that ephedrine into salt. Now that salt is soluble in the water, right? So solubility, you can overcome the solubility problem. So salt formation, most of the organic salts, they are formed by addition or removal of proton from ionized. So most of the salts, they are formed by either addition or removal of proton, you know, to form a ionized drug molecule, and then that ionized molecule is neutralized by counter ion to form a salt. Salt कैसे बनता है? Most of the organic salts, okay, of organic compounds, they are formed by addition of or removal of proton to form a ionized molecule, and then it is neutralized by counter ion to form a salt. Organic salts are more soluble in the water than unionized molecule. You know that salt are more soluble in the water than unionized molecule, because unionized molecule is a lipid soluble, and it is a simple mean to increase the solubility of compound. Salt formation is a simple mean. It is the simplest way to increase the solubility of compound, right? Problems encountered during salt formation. जब salt form बनता है तो उसमें क्या क्या दिक्कत आता है ना? So during the salt formation, the problems like poor crystallinity. तो जो crystal बनेगा वो poor बनेगा. Different degree of solvation, different degree of solvation or hydration, depending upon the nature of, solvent बन जाएगा, hydrate बन जाएगा, monohydrate बन जाएगा, dihydrate बन जाएगा. So जैसे if half water molecule is incorporated during the crystallization, known as a hemihydrate. If one molecule is incorporated into the during the crystallization, known as a monohydrate, trihydrate respectively. Hygroscopicity, instability due to unbearable pH in a crystalline medium. Unbearable pH. Hydrochloride salt, bromide salt, chloride salt, iodide salt, citrate, malate, okay, palmoate, phosphate, sulfate, and tartrate. They are most commonly used anions. है ना? आपने देखा होगा अगर कोई बेसिक ड्रग है तो उसका सॉल्ट बनता है ना, आयरन सिट्रेट, है ना, क्लोरम्फेनिकॉल palmoate, like that, है ना, कोडीन फॉस्फेट, है ना, जैसे आपका मॉर्फिन सल्फेट, है ना, like that. So most commonly for salt formation, anions used are nothing but the hydrochloride, bromide, chloride, iodide, citrate, malate, palmitate, phosphate, sulfate, tartrate. Okay, and for cations, these are the mostly used cations like sodium, potassium, calcium, and zinc. So few examples of drugs and their salt formation is explained over here. So salt forming agent. सॉल्ट किसका बनता है, कौन है, मॉडिफाइड ड्रग कौन सा है, राइट, एंड मॉडिफिकेशन क्यों किया है, व्हाट इज द आउटकम ऑफ मॉडिफिकेशन. सो फॉर एग्जांपल डॉक्सीसाइक्लिन इज द ड्रग, it is a tetracycline. Now it is okay. Doxycycline is modified by using this acetyl amino acetic acid. So salt forming agent is acetyl amino acetic acid. So to increase the solubility of doxycycline, So doxycycline is converted into salt of acetyl amino acetic acid, right? So likewise that erythromycin is converted into salt of N-acetyl L-aspargine to enhance the solubility, okay, again activity and stability. Again doxycycline is formed a salt with the N-acetylcysteine, so it is having combined effect in the pneumonia. Means this salt, N-acetylcysteine and doxycycline, their common salt. Doxycycline will act as an antibacterial agent in pneumonia, and this N-acetylcysteine, इसका काम है कि it will break down the mucus ना in the respiratory tract. So synergistic effect. Then like alkyl bicarbonate. So bicarbonate are antidiabetic drug. They are they are formed salt with the aldamanitic acid, adamanotic acid, adamantoic acid. So to increase the duration of action, to prolong the action, right? So piperazine is formed salt with the this adipic acid salt of piperazine. Now stability of piperazine is increased, toxicity organoleptic toxicity is decreased, organoleptic property is increased. Likewise, that's why salt formation. Then ampicillin, lincomycin, so they are converted into salt of N-alkyl sulfamates to enhance the oral absorption and stability respectively. So this is the example of salt formation. Are you getting? Salt क्यों बनाते हैं? Yes or no? Are you getting? So these are the, am I audible to you? Yes, salt formation to increase the solubility, to increase the stability, to increase the kinetics, etc. etc. Right? Few examples of salt and their salt forming agent and their modified drug. Okay, तो modified drug salt forming agent इसका salt बनते हैं किसके साथ में ये doxycycline acetyl aminoacetic acid salt, erythromycin, N-acetyl asparagine, doxycycline, N-acetylcysteine like that. Piperazine, adipate, adipate, piperazine adipate, piperazine salt of adipic acid like this. Now we'll see the next part. Now again you can see cephalaxine. Okay, cephalaxine is converted. It is formed a salt with the anthraquinone 1,5 disulfonic acid. Anthraquinone 1,5 disulfonic acid के साथ में cephalaxine का salt बनाता है to increase the solubility and absorption rate. वैसे various alkaloids they are formed a salt with the arabinogalactone sulfate to increase the duration of action. Likewise, that cephalosporin, sulfabenzyl, penicillin, they are formed a salt with the arginine to toxicity is decreased and stability is increased. Okay, hygroscopicity, toxicity is respectively. So to decrease this way, then erythromycin is formed salt with the aspartic acid. So erythromycin aspartate to enhance the solubility. Tetracycline, betaine के साथ में tetracycline का salt बनाते हैं. Okay, to increase the gastric absorption, right? So to decrease the toxicity of metformin, metformin is converted, it is formed a salt with the carnitine. Like propaxipine, it is a opioid drug. ये क्या होता है? Opioid analgesic होता है. It is mostly used as a antispasmodic. Okay. Propaxipine is, okay, it's salt with the 4-chloroamptolene sulfonic acid is converted to increase the organoleptic properties. Right. So few example of salt and their respective. Now what do you mean by products? Products आपने देखा है pharmacology में, we already seen. Products, they are nothing but they are inactive drugs as such, are metabolized in the body. They are metabolized in the body, and they to release the active drug. No? Products, they are actually they are known as inactive medicinal agents, and in the body after metabolism they are activated, right? Now products again, few drugs are converted into products. Why? Again to alter the absorption pattern, distribution metabolism pattern, to increase the lipophilicity. So products are formed. Products भी क्यों बनाते हैं? Again लिपोफिलिसिटी इंक्रीज करने के लिए या फिर absorption distribution kinetics enhance करने के लिए, to enhance the kinetics and lipid solubility. Now okay, again pharmaceutical improvement and stabilization, to increase the stabilization, to increase or decrease the solubility, the salinity. Okay, for example, we have a lot of products. For example, यहाँ पे एक example दिया है erythromycin histolate. Erythromycin histolate. Erythromycin histolate is the, am I audible? Am I audible? Erythromycin histolate is the prodrug. It is a water soluble salt of lauryl sulfate. Water, okay. So erythromycin histolate is water insoluble lauryl salt of the propionate ester. Okay, that's why it is known as erythromycin histolate. Histolate क्यों बोलते हैं? Because it is a salt of propionate. It is a ester salt. Okay, it is a ester of propionic acid. Erythromycin plus propionic acid ester with the, okay, lauryl sulfate. That's why erythromycin histolate. Erythromycin histolate. Water insoluble lauryl sulfate salt of propionate ester. That is the erythromycin histolate. Okay, erythromycin histolate. क्यों? इरेथ्रोमाइसिन इन वाटर इज प्रोटोनेटेड. इरेथ्रोमाइसिन जब वाटर में आता है तो प्रोटोनेशन होता है उसका, है ना, पॉजिटिव चार्ज होता है. एंड is a water soluble. Erythromycin in water is protonated, then it is water soluble, but it has a bitter taste and quickly it is hydrolyzed in the GIT, gastric acid, okay, to become an inactive product. जब नॉर्मली इरेथ्रोमाइसिन लेते हैं तो वाटर में उसका प्रोटोनेशन होता है एंड वो उसका बिटर टेस्ट होता है and it is quickly hydrolyzed by acid and converted into inactive product. Right? Now, to overcome this issue, erythromycin is converted into product. Erythromycin histolate. Now, ester and amide. They are the ester and amide products. Ester and amide products are designed to increase the lipophility. Products. Ester and amide products are designed to increase the lipid solubility. Water solubility is also increased by binding with the amino acid. अगर if you want to increase the lipid solubility of drug, तो आपको क्या करना है, वो ड्रग किसके साथ में अटैच करना है, you have to attach that drug either with the amide or ester. So it will increase the lipophilicity of that molecule. Now if you want to increase the water solubility, you can bind that drug with the amino acid. So water solubility will be increased, right? So prodrugs. What is the term solidity? Solidity means it is nothing but the concentration at which, okay, the concentration of drug at which solution phase is in equilibrium with the solid phase at a given temperature. Solubility अपने देखा है, we already seen solubility in the pre-formulation. So it is nothing but the concentration at which solution phase is in equilibrium with the solid phase. Now factors which are considered during the experiment. So while doing the experiment, solubility experiment जब करते हैं उसमें क्या क्या फैक्टर आपको रखना है, कंसिडर करना है. While determining the solubility, the factors you have to consider like pH, temperature, ionic strength, concentration, etc. etc. Now, equilibrium solidity is measured based upon the phase solubility technique by Higuchi and Connors. तो एक बार क्वेश्चन आ चुका है, Higuchi and Connors law is related with what? Is related with the solubility. So जब solubility experiment करते हैं, so these factors you have to take into consideration: pH, temperature, ionic strength, concentration. Now equilibrium solubility is measured based upon the phase solubility technique proposed by Higuchi and Connors. आप बस इतना ही रखो, Higuchi and Connors, they are related with the solubility. इतना याद रखना है. नाइपर में क्वेश्चन आता है आपको. So how to do solubility experiment? How to conduct? If some drug dispersed in the solvent in the container and agitated, some amount of drug is solid is dispersed in the solvent, okay, in a container and it is stirred at a constant temperature for a particular time. Okay, then after a particular time, at a particular time interval, the sample is returned as a function of time. Now that sample is clarified and now it is assayed by either gas chromatography, HPLC, UV, fluorescence spectroscopy to establish the solid from the drug. Okay. Now drug with the solubility less than 1%, जिसका solubility 1% से कम है in the GI tract, it will show poor and erratic absorption. वो जल्दी absorb नहीं होगा. Solubility कैसे मेजर करते हैं? Solubility in the water, aqueous solubility, then organic solubility, organic solvent में solubility उसका मेजर करते हैं. Solubility experiment is also done by using taking a 0.9% sodium chloride ये solubility का experiment कैसे करते हैं? ऐसे करते हैं, some amount of solid you have dispersed in a solvent container and you have to stir it at a constant temperature, and you have to withdraw a samples at a particular interval, and then you have to assay by using gas chromatography, HPLC, UV, fluorescence spectroscopy to establish the equilibrium of a solid form of drug. Right now, अगर कोई ड्रग का solubility लेस देन 1% है इन GIT फ्लूइड, सो वो जो ड्रग है वो बहुत ही पुअर एब्सॉर्प्शन दिखाता है एंड इरेटिक एब्सॉर्प्शन दिखाता है। सोलिबिलिटी कैसे मेजर करते हैं? बाय यूजिंग डिफरेंट डिफरेंट सॉल्वेंट, लाइक जैसे वाटर हो गया या फिर 0.9% सोडियम क्लोराइड हो गया, ऑर्गेनिक सॉल्वेंट हो गया। नेक्स्ट विल सी व्हाट इज द इफेक्ट ऑफ relative humidity on the appearance of flow properties of powder. Yes, anybody has any doubt? Yes, effect of relative humidity on the appearance and flow properties of the powder. किसी कोई डाउट है क्या? Yes. So what is the effect of relative humidity on the flow properties of material or powder? तो जैसे ह्यूमिडिटी बढ़ गया तो फ्लो प्रॉपर्टीज के ऊपर क्या इफेक्ट होगा उसके रिगार्डिंग है। So for example यहाँ पे दिया है, so when relative humidity okay is at a 0%, if relative humidity is 0%, so and at the time moisture content is a 0.31% weight by weight, so at that time powder is a free flowing and it is easily passed through sieve. So है ना, जब रिलेटिव ह्यूमिडिटी 0% होता है तो पाउडर जो है वो फ्री फ्लोइंग होता है एंड वो इजीली सीविंग कर सकते हो। इट कैन बी पास्ड इन द सीव, it can be easily passed through sieve and it is a free flowing at a 0% relative humidity. Now if you increase the relative humidity, so 11.3% say for example, so again water content is a 0.24% and it is again free flowing powder easily passed through sieve. अगर if you increase the relative humidity about 22.5%, water content is called 0.2%, 27 weight by weight. Now it is a less free flowing. Yeah. So effect of percentage of relative humidity on the flow appearance and the flow properties of powder. ह्यूमिडिटी अगर कम हो गया तो मटेरियल का जो अपीयरेंस है उसके ऊपर क्या इफेक्ट है या फिर उसका फ्लो प्रॉपर्टीज के ऊपर क्या इफेक्ट है, दैट्स व्हाई सो एट 0% relative humidity powder is a free flowing easily passed, again at 11.3% also it is also free flowing and easily passed. But if you increase the humidity at 22.5%, so it is a less free flowing. So if you increase the humidity at 38.2%, so again flowing powder is flowing but tends to adhere to the surface of either dye or CO, etc. If relative humidity is increased up to 57.6%, it is a less free flowing. Again if you increase the relative humidity 75.3%, material is being caked, इसका केक बन जाए. Ambient 100% humidity, powders adhere to the surface of petri dish. So that is the effect of relative humidity on the appearance as well as flow properties of powder. बस इतना, यहाँ से कोई क्वेश्चन आएगा इतना कोई नहीं पूछता कि बस याद रखना है कि 0% relative humidity है तो पाउडर जो है मटेरियल जो है वो फ्री फ्लोइंग रहेगा. There is a free flowing, right? Now if you increase the humidity, so obviously that has an effect on the flow properties of powder, right? Now various phases of transformation of a compound, how compound is converted into one way, how compound is converted from one form to another form. जैसे for example crystalline amorphous form होता है. So what type of transformation? Molecular transformation. जैसे आपका सॉल्ट फॉर्म होता है, पेरेंट का ड्रग होता है. सो हाउ देयर इज अ ट्रांसफॉर्मेशन फ्रॉम ए फ्री ड्रग टू द सॉल्ट फॉर्म. हाउ देयर इज अ ट्रांसफॉर्मेशन ऑफ अ ड्रग फ्रॉम हाइड्रेट एंड डिहाइड्रेशन, हाइड्रेट फॉर्म एंड डिहाइड्रेटेड फॉर्म. अनहाइड्रस फॉर्म जो होता है देयर इज अ विदाउट वाटर मॉलिक्यूल, हाइड्रस फॉर्म इज वाटर मॉलिक्यूल. हाउ देयर इज अ ट्रांसफॉर्मेशन बिटवीन हाइड्रस टू अनहाइड्रस or anhydrous to hydrous, polymorphic transfer. So there are different levels of there are different levels, or you can say there are different phases of transformation of compound. So various phases of transformation of compound. ये पार्ट आपको किसी ने पढ़ाया भी नहीं होगा, आपने कहीं देखा भी नहीं होगा. अगर आपने लैकमन पढ़ा है तो उसमें है, लेकिन उतना कोई कॉन्सेंट्रेट नहीं कर. बट मुझे लगता है कि इसके ऊपर क्वेश्चन आ सकता है, दैट्स व्हाई आई हैव एक्सट्रैक्टेड। नाउ टाइप्स ऑफ मॉलिक्यूल, ओके, टाइप्स ऑफ, ओके, फेजेस ऑफ डिफरेंट ट्रांसफॉर्मेशन ऑफ कंपाउंड। सो इसके फेजेस होते हैं फेज ए, फेज बी, सी, ओके, फेज बी, फेज सी, फेज डी, फेज ई, ओके, फेज एफ, इतने फेज होते हैं। सो विल सी वन बाय वन। व्हाट इज फेज वन? Sorry, what is phase A? Phase A is nothing but the known as the polymorphic transitions. So phase A को बोलते हैं polymorphic transition. So okay, the transition occurs between different polymers. Transition between polymorphs, which includes all type of crystalline solid. Okay. Now, while transition एक एक पॉलीमॉर्फ से दूसरे पॉलीमॉर्फ में जब उसका ट्रांजिशन होता है तो वहां पे क्या चेंज होता है? सो सॉलिड कंपोजिशन रिमेन्स सेम। सो इन द phase A or in a phase A transformation, so that is a nothing but the phase A transformation is nothing but the पॉलीमॉर्फिक ट्रांजिशन, है ना। अगर किसी कंपाउंड के दो पॉलीमॉर्फ ए एंड बी, वो ए पॉलीमॉर्फ से बी दूसरे पॉलीमॉर्फ में जाने में जो ट्रांजिशन हो रहा है, ट्रांजिट हो रहा है ना, तो इट इज ट्रांसफरिंग फ्रॉम वन फॉर्म टू अनदर फॉर्म। सो दैट पॉलीमॉर्फिक ट्रांजिशन इज नोन एज अ phase A transformation. Okay? And during the transition, so solid content or solid composition of that compound remains same. तो आपको क्वेश्चन कैसे आएगा? डायरेक्ट क्वेश्चन आ सकता है, ओके, डायरेक्ट। व्हाट इज phase 1 transformation in the compound? That is a polymorphic transition. Okay? Now phase B, so phase B is nothing but the transition between, okay, transition between phase B is known as hydration dehydration transitions. Hydration dehydration transition, you know, it is a transition between anhydrates and hydrates or hydrates of different stoichiometry. एक तो ये ट्रांजिशन किसमें होगा? फेज बी में। फेज बी का जो ट्रांजिशन होता है मॉलिक्यूलर ट्रांजिशन, वो ट्रांसफॉर्मेशन इन द कंपाउंड, फेज ऑफ ट्रांसफॉर्मेशन। फेज ए में होता है पॉलीमॉर्फिक ट्रांजिशन, वन पॉलीमॉर्फ टू अनदर पॉलीमॉर्फ एंड सॉलिड कंटेंट रिमेंस सेम, इतना याद। Phase B of transformation of compound, that is nothing but the transition between hydration and dehydration. Hydration and dehydration, मतलब अगर कोई फॉर्म है हाइड्रेट फॉर्म है या फिर कोई अनहाइड्रेट फॉर्म है, so how a hydrous form will convert into anhydrous form, so that transition is nothing but the phase B of molecular transformation. Okay? तो molecule transform कैसे हो रहा है, तो उसके phases है, right? Are you getting or not? Okay. So okay, so phase B of transformation of compound involve the hydration dehydration. So transition between anhydrates and hydrates, है ना, या फिर transition between hydrates and hydrates of different stoichiometry, है ना, different stoichiometry। अगर 1:1 एडिशन हो रहा है that is a stoichiometric, 1:1 नहीं हो रहा है तो that is non stoichiometric। So hydrates and anhydrates transition between anhydrous form, hydrous form, transition between different hydrous form of different stoichiometry. Okay. In this also solid composition may be different. The solid composition of that solid composition of that hydrate or dehydrate form is a different now. Difference is being number of water molecule. So here solid composition changes in terms of water molecules. Okay? So it differs by number of water molecules. Now नेक्स्ट होता है आपका ट्रांजिशन, होता है ट्रांजिशन सी, फेज, okay, phase C, phase transformation. So we have seen the A, B and C. Now C, so C phase of transformation means what? So C phase of transformation in the compound means सॉल्वेशन डिसोल्वेशन। तो यहाँ पे सॉल्वेशन डिसोल्वेशन, यहाँ पे क्या होता है? ट्रांजिशन बिटवीन द प्री क्रिस्टल फॉर्म एंड सॉल्वेट फॉर्म, प्री क्रिस्टल एंड सॉल्वेट, सॉल्वेशन डिसोल्वेशन। फेज सी ऑफ मॉलिक्यूलर ट्रांसफॉर्मेशन इज नोन एज अ सॉल्वेशन डिसोल्वेशन। आपको डायरेक्टली क्वेश्चन आ सकता है, व्हाट इज द फेज ए ट्रांसफॉर्मेशन इन अ सॉलिड कंपाउंड? सो फेज इज नथिंग बट द ट्रांजिशन बिटवीन पॉलीमॉर्फ्स, है ना, एक पॉलीमॉर्फ से दूसरे पॉलीमॉर्फ्स में जाते समय क्या चेंज हो रहा है या फिर नहीं हो रहा है, दैट इज एक्सप्लेन्ड बाय फेज ए ट्रांसफॉर्मेशन ऑफ कंपाउंड। फेज बी में क्या होता है ना, हाइड्रेशन डिहाइड्रेशन। फेज सी क्या है, सॉल्वेशन डिसॉल्वेशन. सो ट्रांजिशन बिटवीन सॉल्वेट फ्री क्रिस्टल, सॉल्वेंट फ्री क्रिस्टल, क्रिस्टल है उसमें सॉल्वेंट नहीं है, दूसरा एक क्रिस्टल बन गया है जिसमें सॉल्वेट है, मतलब सॉल्वेट मतलब वाटर, सो वाटर सॉल्वेंट ऑफ क्रिस्टलाइजेशन उसमें एंटर हो गया. सो हाउ देयर इज अ फेज चेंज, हाउ देयर इज अ ट्रांजिशन बिटवीन सॉल्वेंट फ्री क्रिस्टल एंड solvent pre-crystal form and solvate form. Okay, that is a phase C transformation or transition between different stoichiometric solvents and different nature of solvents. Okay, in this also solid composition differs by nature and number of solvent molecules. I will see next is nothing but the phase D transformation. What is phase D transformation? So phase D transformation is nothing but the फेज डी ट्रांसफॉर्मेशन ऑफ द कंपाउंड. फेज ए में क्या हो रहा है कि पॉलीमॉर्फिक ट्रांजिशन हो रहा है, फेज बी में क्या हो रहा है कि हाइड्रेशन डिहाइड्रेशन ट्रांजिशन हो रहा है, फेज सी में क्या है सॉल्वेशन डिसॉल्यूशन ट्रांजिशन हो रहा है, फेज डी जो है फेज डी ऑफ ट्रांसफॉर्मेशन ऑफ कंपाउंड उसमें क्या हो रहा है सॉल्ट पेरेंट कन्वर्जन और सॉल्ट सॉल्ट एक्सचेंज. तो इट इज अ ट्रांजिशन बिटवीन सॉल्ट, सॉल्ट फॉर्म मतलब क्या होता है सॉल्ट मीन्स आयनिक एडक्ट, the transition between salt, that is ionic adduct, and parent or unionized free acid or free base, or transition between salt of different stoichiometry or transition between different salt. So in this solid composition differs, there is a difference in the solid composition like number and nature of counter ions. Now solid composition कहाँ पे same है, so in the phase A. Okay, in the phase transformation, phase A of compound transformation. So transformation during phase A, solid composition remains same. There is a transition between polymorphic form. E phase of transformation, phase E of molecular transformation. So it is known as a co-crystal parent conversion or co-crystal exchange. Co-crystal parent conversion or co-crystal exchange. Now in this phase E of molecular or compound transformation, so in this it is a transition between co-crystal मतलब molecular order and parent compound, co-crystal and parent compound. और कोई parent compound क्या है आयोनाइज्ड कंपाउंड है या फिर सॉल्ट है, और molecular order क्या है co-crystal है. Again transition between co-crystal of different stoichiometry or different nature, transition between different nature of crystal. Here again solid composition differs by nature and number of co-crystal form. Okay. So this is known as a different phases of molecular or compound transformation. तो यहाँ पे compound transform कैसे होगा उसके phases है. We also seen this है ना. ये प्री फॉर्मुलेशन में जब अपने देखा है polymorphism and crystallinity. So in that okay. Now mechanism of phase transformation, mechanism of phase transformation. तो ये phase transformation सॉलिड में कैसे होगा, लिक्विड में कैसे होगा, सॉल्यूशन में कैसे होगा. सो इन द सॉलिड कंपाउंड फेज ए एफ ट्रांसफॉर्मेशन होगा, फेज ए टू एफ ट्रांसफॉर्मेशन. इन द ओके सेकंड होता है मेल्ट, मेल्ट में ए फेज ए बी सी, यहाँ पे डिहाइड्रेशन डिसॉल्यूशन ओनली होता है. देन डी ई एफ विट्रिफिकेशन ओनली. तो जब मेल्टिंग होता है कंपाउंड का, so these are the phases transformation observed in the molecule while molecule is melting जब होता है molecule का तो phase A transformation B C तीनों भी phase A B C है ना. C में सिर्फ जो dehydration dissolution होता है सिर्फ ये यहाँ पे heat अप्लाई कर रहे हैं. and then D E F तो all phase all phases transformation that occurs in the melting. सॉल्यूशन में अगर कोई ड्रग सॉल्यूशन में डाले तब कौन से फेजेस ओके कौन सा ट्रांसफॉर्मेशन को. सो इन द सॉल्यूशन phase transformation is between A and F. Okay. Then solution mediated phase transformation, then A E एक होता है, then F होता है. हमारे पास क्रिस्टलाइजेशन अपने फेज F जो है ये बाकी है. फेज F क्या होता है, फेज F ट्रांसफॉर्मेशन में It is a transition between amorphous crystallization or vitrification. So transition between crystalline and amorphous form. Composition of solid again यहाँ पे change होगा. Changes in the most of the cases. So these are the different phases of transformation. Phase A, B, C, D, E and F. So याद रखना है ये phase. So amorphous crystalline transition किस में होता है? Phase F में होता है, याद रखना. Phase F, ठीक है. एक बार रिवाइज करो फिर से. Phase A of compound transformation. That is nothing but the transition between polymorphic with different polymorph. Polymorph के बीच में जो changes हो रहे हैं, one polymorph is converted into another polymorph. So that transition is given by phase A transformation of molecule. Phase B hydration dehydration जो होता है ना, so that is given by phase B transformation. Phase C transformation मतलब क्या है ना, solvation desolvation, that is phase C. Phase D क्या है, salt parent conversion और salt salt action. Phase E क्या होता है, co-crystal parent conversion और co-crystal action. और phase F क्या होता है, phase F is nothing but the amorphous crystalline transition और vitrification. सो आर यू गेटिंग और नॉट, समझ रहा है आपको यहाँ पे, क्योंकि ये किसी ने पढ़ाया भी नहीं होगा आपको, ना किसी को मालूम भी है. आर यू गेटिंग और नॉट, है ना, ना आपके कॉलेज में पढ़ा है, गारंटी के साथ बोलता हूँ. ये आप उनको भी पूछो, आपके स्टूडेंट्स से जो टीचर होते हैं उनको भी पूछो. ये नेक्स्ट पार्ट इज नथिंग बट द different biopharmaceutical consideration while designing a dosage form. बायोफार्मास्यूटिक्स के कौन से कंसिडरेशन करना है आपको जब टैबलेट बनाते हो, कैप्सूल बनाते हो, डोसेज फॉर्म बनाते हो. बायोफार्मास्यूटिकल कंसिडरेशन ऑफ ड्रग प्रोडक्ट डिजाइन. पहला है आपका एपीआई. एपीआई मींस योर ओन ड्रग. एपीआई मींस एक्टिव फार्मास्यूटिकल इंग्रेडिएंट और ड्रग. सो यू हैव टू टेक कंसिडरेशन, you have to take stability of API into consideration, then solidity, then pH, pKa, polymorphism, drug excipient interaction, and drug excipient compatibility. So about the drug, you have to take these things into consideration while designing a dosage form. तो आपको ड्रग के बारे में क्या क्या कंसिडर करना है, यू हैव टू कंसिडर स्टेबिलिटी ऑफ ड्रग, you have to consider solubility of drug, pH, polymorphism, pKa, drug excipient interaction, and drug excipient compatibility. So while designing a dosage form, you have to take these things into consideration while about the drug. Then impurities, salt form, particle size, and complexation. These things also you have to take into consideration. Now, so these things you have to take consideration for the drug. Now for the product product for the product consideration, about the product type of dosage form you have to take into consideration, conventional or modified release, then dose strength, bioavailability, excipient stability, and manufacturing variables. You have to take these things into consideration while designing a dosage form which are product related. ड्रग प्रोडक्ट रिलेटेड ये फैक्टर्स है, एपीआई के रिलेटेड ये पॉइंट्स है. वैसे फिजियोलॉजिकल फैक्टर्स कौन से है, अगेन रूट ऑफ एडमिनिस्ट्रेशन, परमेबिलिटी ऑफ ड्रग, प्रोटीन बाइंडिंग ऑफ ड्रग, ब्लड फ्लो, मेटाबॉलिज्म, काइनेटिक्स सिस्टम. फार्माकोकाइनेटिक्स एंड फार्माकोडायनामिक्स कंसिडरेशन कौन से है, ऑब्वियसली बायो अवेलेबिलिटी, एडवर्स ड्रग इफेक्ट, थेरेपेटिक ऑब्जेक्शन if any, टॉक्सिक इफेक्ट if any. मैन्युफैक्चरिंग कंसिडरेशन कौन से लेने हैं भाई, while designing a dosage form you have to consider these things: production methodology, quality control, quality assurance, raw material specification, cost and stability testing. ये मैन्युफैक्चरिंग रिलेटेड पॉइंट्स कंसिडर करना है. पेशेंट कंसिडरेशन कौन से करना है, कंप्लायंस, लेबलिंग, प्रोडक्ट एक्सेप्टेंस एंड कॉस्ट. दीज आर द पेशेंट रिलेटेड considerations or patient related factors. So what are the different factors you have to take into consideration while designing a dosage form? So API related factors कौन से, stability, solubility, pH, pKa, polymorphism, excipient drug excipient interaction, compatibility, impurity, salt formation, particle size and composition. वैसे ही ड्रग प्रोडक्ट के रिगार्डिंग कौन से फैक्टर्स है, टाइप ऑफ डोसेज फॉर्म, either conventional or modified, dosage strength, bioavailability, excipient stability and manufacturing variables. ये प्रोडक्ट रिलेटेड है, right? So product related consideration. Now manufacturing related consideration means a production methodology is there, quality control, quality assurance, raw material specifications क्या है, cost and stability listing. Now physiological factors कौन से consideration कौन से, route of administration, permeability of drug, protein binding, blood flow, then metabolism, kinetics. Pharmacokinetics and dynamic consideration कौन से, bioavailability, adverse effect, therapeutic objection, even toxic effect. पेशेंट रिलेटेड फैक्टर कौन से, कंप्लायंस, लेवलिंग एंड प्रोडक्ट एक्सेप्टेंस, कॉस्ट, ये फैक्टर्स है. सो व्हाइल डिजाइनिंग द डोसेज फॉर्म यू हैव टू कंसीडर दिस फैक्टर्स. व्हाइल डिजाइनिंग ए डोसेज फॉर्म यू हैव टू कंसीडर these factors, right? How drug is, okay, how drug is introduced, how it is liberated, how it is absorbed. So first it is given by this system known as a Latimer system. What is Latimer system? These are the capital letters L, A, D, M, E, R. L मतलब क्या, liberation, A मतलब absorption, distribution, metabolism, elimination, इसको बोलते हैं response. Lastly है response. So Latimer. So if you take a tablet, आपके स्टमक में टेबलेट जाएगा, वहाँ पे वो डिसइंटीग्रेट होगा, है ना, उसके बाद में डिसोल्यूशन होगा, डिसोल्यूशन एंड ड्रग रिलीज मतलब लिबरेशन. After that there is an absorption of drug. So absorption of drug in systemic circulation. उसके बाद में मेटाबॉलिज्म होगा, वहाँ पे एलिमिनेशन होगा, मेटाबॉलिज्म एंड एक्सक्रीशन. तो absorption के बाद में डिस्ट्रीब्यूशन भी होता है, डिस्ट्रीब्यूशन ऑफ ड्रग इन द बॉडी fluids, so drug in the tissue or in the plasma, and then response, then response. तो सीक्वेंस कैसे होता है, लिबरेशन ऑफ ड्रग, then absorption of drug, then distribution of drug, then metabolism, elimination and response. So this is known as a, this happens with drug. ड्रग के साथ में क्या क्या होता है ना, ड्रग के साथ में क्या होता है, फॉर्मुलेशन जब आप लेते हो कोई टैबलेट कैप्सूल, so drug will be liberated, it will be absorbed, it will be distributed, it will be metabolized, it will be eliminated, and it may show the response. This is known as a Latimer system. Say granules, say for example आपने टैबलेट ले लिया, टैबलेट और कैप्सूल. So when you take tablet or capsule, it will go in the GIT, either it will disintegrate. Disintegrate होगा मतलब क्या, tablet is converted into small particles, that is a disintegration. So granules all aggregates, but if it is a fast soluble, then directly dissolution will be there. Drug in a solution ultimately. Drug in a solution, either it may be in an ionic form, that is ionized form, which is not able to cross the biological membrane. Ionized अगर drug है तो it is not able to cross the biological membrane. Non-ionic form मतलब non-ionized drug or unionized drug. Unionized drug. Now ionized drug or ionized form is a water soluble. Unionized form of drug is a lipid soluble. It is easily get absorbed in lumen GIT. That is a summary of process involved in the systemic drug absorption after oral administration. We have already seen in the pharmacology tutorial. Now transport mechanism, how drug is absorbed, different transport mechanisms are there, like passive diffusion, all that we already seen. Now transport of drug, transport of drug molecule through a cell is known as a paracellular transport. It's known as a paracellular. हाँ मैं बोला,
हाँ सर 500 तक रीचिंग हुआ सर.
ठीक है, मैं चेक करके बताता हूँ आपको कहाँ पे.
हाँ सर स्टेटस बता. ओके सर.
There is also paracellular transport of drugs across the cell, through the cell, known as a paracellular. Then transcellular, between the cell, transcellular. Then protein mediated transport, are proteins are nothing but a carrier. These carrier, they will bind to a drug and then it is transported across the cell membrane. So for example paracellular transport is applicable for hydrophilic substance जो water soluble है उसके लिए, through the cell, between the pores of cell. Now transcellular transport mechanism is applicable for lipophilic substances. Okay, and this protein transports or protein mediated transport is applicable for this glucose, amino acids. Receptor mediated, so like it is like insulin, hormones, etc. Absorb to endocytosis or exocytosis, that is for the example albumin and other plasma proteins. Okay. Then pumps are there on a cellular membrane, efflux pumps, which will remove a drug molecule out of the like that. So efflux pumps, one एक अंदर जाएगा और एक बाहर जाएगा like that. So we have seen earlier. Drugs and xenobiotics they are absorbed by efflux pumps. Now absorption of drug in the GIT. GIT में कहाँ से कहाँ पे absorption. So anatomical part of your GIT, respective function दिया है उसका, and effect on a drug absorption. So oral cavity. So oral cavity का pH कितना होता है? Question आ सकता है आपको. What is the pH of oral cavity? 7. pH of oral cavity is 7. Now in the oral cavity there is a one enzyme known as taline. इसके ऊपर क्वेश्चन आ चुका है. There is a this enzyme will digest the carbohydrate. Taline from saliva digests the starch. So if you take a chapati and if you try to chew in the mouth, गरम गरम चपाती ले लो, उसको थोड़ा सा मुंह में चबाओ बहुत देर तक, तो आपको एक स्वीट टेस्ट आएगा. Why? Because you have saliva. In that saliva there is one enzyme known as taline, which will digest the starch and convert into disaccharide, which is a sweet in taste. Okay. Now in the oral cavity, starch is digested by taline. Then mucin is also there in the oral cavity, which will lubricate the food. Now this mucin may interact with the drug. Effect on a drug absorption. यहाँ पे buccal and sublingual absorption takes place for lipid soluble drug. Or esophagus, the pH of esophagus is around 5 to 6, and what is the role? It prevents the acid reflux from stomach to the mouth. Very small drug distribution. Very small drug distribution. Small, okay. Then आता है आपका stomach at a fasting state. What is the pH at a fasting? 1 to 3, and when stomach is full, 4 to 5. Now in the stomach acid is secreted. Acid secretion that is stimulated by gastrin and histamine and other things. But acetylcholine also. What it do? It will break down the large food particle. Now, absorption is not efficient in case of basic drugs. यहाँ पे weakly acidic drug ही absorb होंगे because of their they are solubilized in the acid rapidly. क्योंकि basic है acidic है तो उसका ionization होगा. Even the ionized form of drug is poorly soluble in the, it is not absorbed. वैसे duodenum में, what is the duodenum? Okay, in this duodenum enzymatic digestion of protein, because यहाँ पे peptides, यहाँ पे enzyme होते हैं trypsin, chymotrypsin and carboxypeptidase, which will break down the protein, lipids, carbohydrate into monomers for absorption. The main site for the absorption is nothing but the duodenum. Main site. It offers a huge surface area for passive diffusion. Esters products they are hydrolyzed. Bile helps to absorb the lipid solubility. है ना, यहाँ पे this is in the stomach. Drugs may be degraded by acid, or it may be because of the enzymes present in the, ये part जो होता है वो duodenum होता है. So drug may be degraded in the acid, or it may be degraded by the enzyme present in the GIT, and if some, okay, it may be degraded by bacterial flora also. So unabsorbed drug will be passed through feces. Now whatever absorption is there, so that will be absorbed by these blood vessels. Now these are carried and by the portal vein to a liver, and यहाँ पे systemic circulation बाद में जाएगा. So a part of drug which is metabolized in a liver before reaching to a systemic circulation. Now, this type of metabolism is known as a first pass metabolism or pre-systemic metabolism, which is very huge, or it is higher in case of class 1 drug as per the Viapor's prescription. Now jejunum has a pH of 6.5 to 7. So it is there where digestion of protein occurs, carbohydrate, and it is used for in vivo drug absorption study. Most of the drugs are absorbed by passive diffusion over here. Jejunum में passive diffusion से होता है. Ileum का pH होता है 7. There is a less absorption than duodenum. Drug absorbed by passive diffusion over here. Colon का pH होता है आपको 5.5 to 7. In the colon you are having aerobic and anaerobic bacteria which can metabolize the drug. So यहाँ पे कोई विलाय माइक्रोविलाय नहीं होता है. So villi macrovilli, they are present in the jejunum and ileum part. Now in the colon there is limited absorption. Drug like theophylline, metoprolol, okay, they are the good drug for sustained release resistance because they are absorbed in the rectum का page होता है 7 is a 15 centimeter long and end with the anus supplied by superior and inferior hemorrhoidal vein. Now, wherever drug absorption takes place in this suppository enema, they are placed into rectum, which will move directly into systemic circulation if it is near to the superior hemorrhoidal vein. Superior hemorrhoidal, so it is near to the inferior hemorrhoidal vein. We already seen this in the pharmacology. Are you getting my voice properly? Are you okay? Biotransformation मतलब क्या metabolism? Alteration of drug in the body is known as a biotransformation. Bio means living and transformation is alteration. So alteration of drug in the biological system known as a biotransformation or simply metabolism or detoxification. Now it is carried out in a different phases. Okay, phase one. Okay, phase one in that you are oxidation reduction and hydrolysis. ओके, सो यहाँ पे याद रखना है मोस्ट कॉमन फेज वन रिएक्शन होता है आपका ऑक्सीडेशन. क्वेश्चन इसके ऊपर आ चुका है. सो ऑक्सीडेशन इज डन पर जैसे फॉर एग्जांपल वैल्प्रिक एसिड आइबुप्रोपेन दे आर ऑक्सीडाइज्ड. Reduction is done in case of methadone, halothane, etc. Hydrolysis is there in case of aspirin, lidocaine. Phase second reaction known as a conjugation reaction. Phase one reaction known as a non-conjugation reaction. Oxidation, reduction, hydrolysis. What is the purpose of phase one? यहाँ पे purpose याद रखना है to impart polar groups to the molecule. To impart polar groups to the molecule. तो ऑक्सीडेशन होने के बाद में रिडक्शन हाइड्रोलाइसिस होने के बाद में यहाँ पे OH ग्रुप, ओके, COH ग्रुप, SO2 ग्रुप ये ऐड किए जाएंगे ताकि वो मॉलिक्यूल पोलर बन सके टू रिमूव बॉडी. सो aim of phase first is nothing but to introduce a polar group in the molecule so that molecule will become a polar in nature, water soluble in nature, right? What is phase second metabolism? They are known as a conjugation reactions. They are known as a conjugation reactions or synthetic reaction. It is known as a non-synthetic reaction. Okay. Now in this you have glucuronide conjugation, sulfonation, methylation, acetylation, you know, then amino acid conjugation, etc. Acetylation. So salicylic acid, phenopropane, they are glucuronide conjugated. Sulfation of paracetamol, salbutamol is taking place. Methylation in case of morphine, isoprylene. Acetylation in case of hydrazine and isoniazide. You know? But remember there is a phase 3 metabolism also. Mostly यहाँ पे phase दिया है ना 2 phase. Most of the books have given phase 2. बहुत सारे books में सिर्फ यही दिया है कि phase 1 and phase 2. phase 1 में मोस्ट कॉमन होता है ऑक्सीडेशन, फेज 2 में मोस्ट कॉमन होता है ग्लूकोरोनाइड कॉन्जुगेशन. बट फेज 3 भी होता है. फेज 3 क्या होता है? फेज 3 मेटाबॉलिज्म इज नथिंग बट द ग्लूटाथायोन कॉन्जुगेशन. याद रखना है आपको, ये सिर्फ मैं बोल रहा हूँ, पूरे इंडिया में मालूम नहीं किसी को. फेज 3 मेटाबॉलिज्म इज नोन एज अ ग्लूटाथायोन कॉन्जुगेशन. इट इज मेटाबोलाइज्ड, पैरासिटामोल इज मेटाबोलाइज्ड ग्लूटाथायोन कॉन्जुगेशन. सो ग्लूटाथायोन कॉन्जुगेशन इज फेज 3 मेटाबॉलिज्म. ये हो चुका है इसलिए थोड़ा सा पास ले रहा हूँ, ऑलरेडी बहुत अच्छे से हुआ है. नाउ कलर्स यूज्ड इन द फार्मास्यूटिकल. कलर्स आर यूज्ड इन द फॉर्मुलेशन टू एनहांस द its appearance, to enhance the aesthetic value. Now colors, whatever colors we are using in the formulation, so it may be either natural color or semi-synthetic or synthetic color. Now colors in the pharmaceutical, natural color से naturally ये वाले. So source क्या है उसका? Source of this 6,6 dipromo indigo. 6,6 dipromo indigo, also known as a tyrene purple. Tyrene purple color है indigo. So it is obtained from the air oxidation secretion from the glands of the snail. Snail का नाम है Murex brandaris. एक snail होता है, उसमें जो secretion होता है, वो secretion होने के बाद में वहाँ पे air oxidation होके एक compound होता है 6,6 diamomo indigo tyrene. 6,6 diamomo indigo, which is also known as a tyrene purple, right? Now again you can see over here. So this cochineal consists of carminic acid having bright red coloring principle. उसका नेचर होता है एंथ्राक्विनोन. ओके, सो दिस इज प्रेजेंट इन द इंसेक्ट. इंसेक्ट जो होता है, इंसेक्ट ऑकर्स एक्टिव क्रोकस कैक्टी इसमें होता है। एलिजारिन नाम का एक नेचुरल डाई है, दैट इज प्रेजेंट इन द मडार प्लांट। एनेंटिन्स आर प्रेजेंट इन द एनेंटोसिड्स, है ना। देन कैरोट में आता है बीटा कैरोटीन व्हिच विल गिव यू येलो कलर। ब्लू पिगमेंट्स आर फाउंड इन द इंडिगो प्लांट। मिनरल्स में कलर कौन सा आता है? टाइटेनियम डाइऑक्साइड, ओके। रेड एंड येलो फेरिक ऑक्साइड is there, carbon black is there, red ferric oxide, yellow ferric oxide and carbon black. ये mineral color है, these are the colors, some natural colors, ये सब natural है, so they are obtained from natural sources. Now all synthetic, okay, all synthetic colors approved for the use in the pharmaceuticals or drug or cosmetics or food. All synthetic color approved for use must meet the specifications, use and restrictions as described in the title 21st, title 21st or section 21st of CFR, code for federal regulation, part 74, 81, 82. Food drug cosmetic 1938. Now they made a certification for the synthetic colorant, that is mandatory. So if you want to use a color in the formulation, so you have to take certificate from the authority. So the specifications for color is given in the 21 part of CFR code for federal regulation, part 74, 81 and 82. यहाँ पे याद रखना है, इसको क्वेश्चन आ सकता है। Now they have divided color into three category. पहला कैटेगरी होता है food drug and cosmetic. Food drug and cosmetic has divided color into three categories. Permitted for those color which are permitted for food, drug and cosmetic, they are known as F, D and C. Those drugs which are permitted to use in the drug and cosmetic, they are known as D and C. Those D and C. Color for food is also different, or every color is designated by 3 letters number. To separate their category, to distinguish the color between which is used in the food or drug और कॉस्मेटिक। कलर जो यूज करते हैं अपन कलरेंट, उसको 3 डिजिट का नंबर दिया है, उसको एक कैटेगरी दिया है। सो अकॉर्डिंग टू दैट अगर वो कलर फूड में भी यूज कर सकते हैं, ड्रग में कर सकते हैं, कॉस्मेटिक में भी कर सकते हैं, उसकी कैटेगरी बनाई है कलर F, D एंड C। अगर सिर्फ ड्रग एंड कॉस्मेटिक में यूज कर सकते हैं, उसका कैटेगरी बनाया है D एंड C। फक्त फूड है तो फूड F, like this. They have designated in 3 numbers to distinguish these colors from those used in the textile and other function. कलर सिर्फ आपका ड्रग्स में नहीं यूज होता है, कलर सिर्फ आपका सिर्फ फूड में नहीं होता है, कॉस्मेटिक नहीं होता है। ड्रग इज also used in the textile and other uses are also there. So to distinguish your FDC, there is food, drug and cosmetic and food color from the textile and other colors. So there is a 3 numbers designation is one. Certified soluble dyes, water soluble synthetic organic molecules, they are purified intermediate from petrochemicals. They are available in the powder form or granular form or liquid form and dispersion or paste. They are known as certified soluble dyes. Few permanently listed color additives, बहुत सारे हैं। If you want to go, you can read from Lackman and page number is 829. I have just picked one chart, okay, and important from that. तो यहाँ पे 3 चार्ट दिए उन्होंने। Am I audible to you? Am I audible to you? तो वहाँ पे उन्होंने 3 चार्ट, yes, वहाँ पे उन्होंने they have mentioned 3 charts over there, okay, but I have only pick one and उसमें भी important लिए, बाकी सब लेने की जरूरत नहीं है, okay? So color जैसे फॉर एग्जाम्पल FD फूड ड्रग एंड कॉस्मेटिक ब्लू वन जो है इज नोन एज अ ब्रिलियंट ब्लू FCF 42090, ऐसे वैसे ड्रग एंड कॉस्मेटिक ब्लू 4 known as अल्फा, okay, अल्फा जुरिन FG 42090, वो नंबर है। ड्रग एंड कॉस्मेटिक ब्लू 9 है ना, इंडियन थ्रीन ब्लू। फूड ड्रग एंड कॉस्मेटिक ग्रीन 3 known as a fast green FCF. Drug and cosmetic green 6 known as a quinazarine green. Drug and cosmetic ऑरेंज 5 उसको बोलते हैं डायब्रोमो फ्लोरेसेंस। फूड एंड ड्रग कॉस्मेटिक रेड 3 नोन एज इरिथ्रोसाइन, ये याद रखना है, ये DC रेड 3 इरिथ्रोसाइन, ये ओके। फूड ड्रग एंड कॉस्मेटिक रेड 22 नोन एज ईओसिन, ये फूड ड्रग एंड कॉस्मेटिक येलो नोन एज टार्ट्रेजिन येलो। बस एक आधा क्वेश्चन आ सकता है आपको यहाँ पे, तो आ भी नहीं सकता है। लाइक ये फूड ड्रग एंड कॉस्मेटिक ब्लू टू कौन है भाई? इंडिकोटीन। इंडिकोटीन इज नथिंग बट ये फूड ड्रग एंड कॉस्मेटिक ब्लू 2, ड्रग एंड कॉस्मेटिक ब्लू 6 है इंड्यू, ड्रग कॉस्मेटिक ब्राउन 1 है रिसोर्सिन, ड्रग एंड कॉस्मेटिक ग्रीन 5 alizarin cyanide green. These colors are permitted. Drug and cosmetic green 8 pyrazine concentrated. Drug and cosmetic orange 4 orange 2। एक कलर के नाम है यहाँ पे, these colors are used, okay? तो आपको एक दो कलर इसमें याद रखना है, टाट्राजीन जैसे इरिथ्रोसिन, टाट्राजीन इरिथ्रोसिन, क्यूसिन, क्योंकि ये कॉमनली यूज होता है, उनका आपको मालूम होना चाहिए। जैसे इंडिगो, ये ड्रग्स मालूम, ये कलर आपको मालूम होना चाहिए। नाउ फ्लेवर्स, okay, flavor that is added to a preparation to increase the palatability, to increase the compliance, है ना। अगर आपको कोई सिरप है, आपको सिरप अगर वेनिला फ्लेवर का है तो आप अच्छे से पियेंगे उसको। सो फ्लेवर्स आर यूज टू मेडिसिन palatable to improve the taste, filling factor and other factors. Taste is nothing but the sensation or stimulus felt by taste bud। टेस्ट वर्ड में जो स्टिमुलस फेल्ट होता है उसको अपन टेस्ट बोलते हैं। नाउ टेस्ट कैन बी फेल्ट इन दिस वे, लाइक आपको ये टेस्ट मिलते हैं, आपको you will get these kind of taste like sweet, sour, bitter, salty and umami, okay। Feeling factor is nothing but the okay, numbness, then cooling, burning, these are the sensation। फील क्या होता है आपको, जो टंग होता है उसके ऊपर टेस्ट बड होता है, वहां पे फील क्या होता है, नमनेस होता है, कूलिंग होता है या फिर बर्निंग होता है। दे से दिस सेंसेशन दे आर कन्वर्टेड इनटू डिफरेंट टेस्ट, स्वीट, सोर, बिटर, टेस्ट। दे ऑकर्स व्हेन नर्व एंडिंग्स आर स्टिमुलेटेड। फ्लेवरिंग एजेंट में भी either नेचुरल, आर्टिफिशियल और आर्टिफिशियल एंड नेचुरल। जो फ्लेवरिंग एजेंट होता है, इन द फ्लेवरिंग एजेंट ओनली 10% aroma is there and remaining 90% is a carrier only, okay। So aroma or okay, the principle or aromatic principle they are adsorb on the carrier। choice of carrier depends on the physical form of flavor। dry flavor है आपका, so crystalline vanillin you have to use, cinnamon powder you have to use, dried lemon fluid extract, है ना। और aroma plated on the maltodextrin, modified starches, textures, gum arabic। अगर आपका लिक्विड है, फॉर लिक्विड वोलेटाइल ऑयल्स, फ्लूइड एक्सट्रैक्ट, टिंचर्स एंड डिस्टिलेट, प्रोपिलीन ग्लाइकोल, इथाइल अल्कोहल, इसके ऊपर ये लिक्विड होता है फ्लेवर। नाउ फॉर एग्जांपल यहाँ पे देखो अनीस जो है, इट इज ऑब्टेन्ड फ्रॉम पिम्पीनेला अनीसम अम्बिलिफेरी फैमिली। तो अनीस फ्लेवर इज यूज्ड इन द कॉन्सेंट्रेशन ऑफ 300 ppm। कार्डामम जो है, कार्डामम इलायची, इलेटेरिया कार्डामम जिंजिबेरेसी फैमिली, so you have to use in the 5 to 50 ppm concentration। लेमन आता है सिट्रस लेमोनम रोटेसी फैमिली 1000 to 10,000 ppm। वाइल्ड चिली का आता है, वाइल्ड चिली इज ऑब्टेन्ड फ्रॉम प्रोनस सेरोटिनियाना फैमिली रोटेसी, इट इज यूज्ड इन द 50 to 800 ppm। ऑरेंज बिटर सिट्रस औरेंटिनम रोटेसी फैमिली up to 500 ppm you can use। so they are the natural, okay। ऑरेंज स्वीट में सिट्रस साइनेसिस रोटेसी, इसका concentration वेरी होता है। now selection of flavor depends on the different or selection of flavor में भी either impact, okay, impact no, depending on that compound में भी classified into 3 category। impact compounds, second होता है contributory compounds and third होता है differential compounds। मतलब फ्लेवर क्या करेंगे, impact flavors or impact flavoring compounds they shows most of the organoleptic potency। मतलब जो फ्लेवर है वही का फ्लेवर रहेगा, which shows most of the organoleptic potency of flavor। एग्जांपल मिथाइल एथेनोलेट, कॉन्क्रोइड ग्रेप्स। तो ये जो फ्लेवर है वही कंपाउंड को फ्लेवर रहेगा। जो ओरिजिनल फ्लेवर है वही रहेगा. Then contributory compounds में it's they increases the complexity and identity of named flavor. जब ये ऐड करेंगे तो जो फ्लेवर आपने ऐड किया है उसका कॉम्प्लेक्सिटी बढ़ाते हैं, उसका आइडेंटिटी बढ़ाते हैं कॉन्ट्रीब्यूटरी कंपाउंड्स. थर्ड होता है डिफरेंशियल कंपाउंड. सो दे इम्पार्ट अ डिस्टिंक्टिव कैरेक्टरिस्टिक ऑफ इंडिविजुअल फ्लेवर. आपने ऐड किया वैनिला वो प्रोडक्ट में, लेकिन वो प्रोडक्ट वैनिला फ्लेवर से नहीं लग रहा है, दूसरा अलग ही कुछ फ्लेवर आ रहा है उसको. दैट इज नोन एज अ डिफरेंशियल. मिथाइल सैलिसिलेट, ग्लिसरीन, वेनिलिन, हनी, लेमन ऑयल, दे विल एक्ट एज अ डिफरेंशियल कंपाउंड. आर यू गेटिंग और नॉट? समझ रहे हैं आपको नहीं? आर यू गेटिंग और नॉट? हाँ, नहीं तो फिर अपन है ना, ये इसके ऊपर क्वेश्चन आ चुका है ऑलरेडी. फ्लेवर्स इन द प्रेस्क्रिप्शन दे आर grouped into 4 टाइप ऑफ टेस्ट दैट टू बी मास्क्ड. कौन सा फ्लेवर कौन सी टेस्ट मास्क करता है, इसके ऊपर क्वेश्चन आ चुके हैं ऑलरेडी. ओके, फ्लेवर्स इन द प्रेस्क्रिप्शन दे आर grouped into 4 टाइप्स टू बी मास्क्ड. कौन सी टेस्ट मास्क करता है? सॉल्टी टेस्ट अगर है कोई प्रोडक्ट का, इफ यू वांट टू मास्क द सॉल्टी टेस्ट, if you want to mask the salt test, drug like sodium salicylate, ammonium chloride, ferric ammonium citrate. Okay, so these drugs they are salt test. Cinnamon syrup is a best vehicle. Cinnamon syrup is a best vehicle. Orange syrup, citric acid syrup, cherry syrup, coca syrup, wild cherry syrup, raspberry syrup, glycerizer syrup. So they can mask the salty taste. If you want to mask the bitter taste, अगर कोई ड्रग का bitter taste है, जैसे फॉर एग्जाम्पल एंटीमलेरियल. Now antimalarial drugs can be masked with the raspberry, coca syrup, then cherry syrup, then cinnamon syrup, citric acid syrup, licorice syrup, cherry syrup, orange syrup in a descending order of usefulness. So bitter taste है तो वो कौन मास्क करता है? कोका सिरप, raspberry syrup ये मास्क करता है. If your taste is sour or acrid, so raspberry syrup and other food syrup they are useful. Food नहीं यहाँ पे fruit है. Raspberry syrup and other fruit syrup are useful to mask the taste. Okay. For example, ACL. Acrid taste of capsicum is also marked by acacia syrup. अगर taste oily है, ये product is oily. So, for example, cod liver oil taste. Now, it can be masked by adding of wintergreen or peppermint oil. Lemon, orange, anise oil can be useful. For castor oil, if you want to mask, it is emulsified with equal volume of aromatic ओके रोबार्ब सिर्फ. ये टेस्ट मास्क हो जाए. नहीं, कल भी देखेंगे, थोड़ा सा रिविजन कर लूँगा. सो आई थिंक टुडे विल स्टॉप हियर.